The next type of biological macromolecules that we'll be talking about are the proteins. The diverse functions of proteins, aside from being the structure of living systems, is mainly due to their diverse forms. Just like carbohydrates, proteins are actually polymers. So particularly, their macromolecules are known as polypeptides, and their subunits are called amino acids. Amino acids are called as such because the general structure of an amino acid involves the presence of an amino group, which is the basic side of the amino acid, and a carboxyl group, which is the acidic side of the amino acid. And then all amino acids also have an R chain, also known as a side chain. The side chain actually give the uniqueness to each of the 20 or so different types of amino acids. So you can already imagine that there are 20 types of amino acids, and if a particular polypeptide or protein is, let's say in this example, 1, 2, 3, 4, 5 amino acids long, how many varieties can that particular protein take? So if we use mathematics, we know that each one of these places for the amino acids, there are 20 possible amino acids. And then there are 5 amino acid places that we are going to fill because this is a sequence of 5 amino acids. So that's already 20 to the power of 5 varieties for a 5 amino acid long polypeptide alone. So this is another view, a ball and stick model of an amino acid. Amino acids may bind with other amino acids, again through a process called dehydration. And in a process, a bond called a peptide bond is formed. And when several amino acids are bonded together through a peptide bond, a polypeptide is formed. And you can see here the side chains or the R groups are actually indicated in green, and the bonds are also indicated in green lines. And notice how each of the amino acid here contains an R group or a side chain. These side chains, which I mentioned a while ago, actually are the reasons why there are 20 types of amino acids. So again, the carboxyl portion and the amino portion of amino acids are already in place, but the R chain or the side chain may be among the 20 different side chains that are present for amino acids. So there are side chains that are polar in nature, as shown here in red. Okay? So the polar nature of the side chains of these amino acids here give these amino acids their characteristics. Okay? So these are water-loving amino acids, plus their polar side chains are charged. Okay, so there's a charge, an ionic charge going on with these side chains. Then there are amino acids whose side chains are polar but uncharged, as seen here. And then there are amino acids whose side chains are non-polar. And finally, amino acids whose side chains have unique properties. So I'd like to particularly focus on cysteine, which has a sulfur atom in its side chain. So cysteine is actually an amino acid that when it's part of a polypeptide chain, it can cause the formation of disulfide linkages. Okay? So as we see here, sulfur-sulfur linkages. Cysteine is actually present in the proteins of the hair, in keratin. Okay? Especially if you have curly or wavy hair. So, if you go to a hair salon, for example, and say you have a curly hair and you want your hair to be straight, to be straightened, okay? The worker there in the hair salon will actually apply a reducing substance onto your hair, okay, and then let it cure for several minutes, even up to an hour. And as you can see here, the direction of the reaction when there's a reducing substance is going to the left, breaking the disulfide linkages in the proteins of your hair and allowing your hair to straighten as shown in the picture here on the left. Okay? So pag nagpa-straight kayo ng buhok, gagamitan ng reducing chemical substance. Likewise, if you want your hair to be wavy or curly because you have a straight hair, okay, the person there at the salon will use an oxidizing substance to oxidize the side chains of the cysteine residues in the polypeptides found in your hair, causing the formation of disulfide linkages. And this causes your hair to become wavy or curly. Amino acids also behave differently in certain types of environments. So glutamic acid, for example, is in the ionized form, its side chain is in the ionized form in basic environments, while lysine is in ionized form in acidic environments. The physiological pH of the human body is around 7.4, so slightly basic. So our glutamic acid is most likely in its ionized form inside our bodies. So, because proteins are made of a sequence of amino acids, and the structure of proteins do not end there, we may actually classify the levels of structure of these protein molecules. So the most obvious one is the primary structure, and this simply has to do with the sequence of the amino acids that are linked in peptide bonds in the protein. Okay? So remember, your protein or polypeptide is made through a process called translation in the ribosomes. Okay? And the first portion or the first end that comes out of that translation process is called the N-terminus of your polypeptide. Okay, so that's this portion, the N-terminus, which ends with the amino portion of the amino acid. And the last portion that comes out of the protein or the polypeptide after it's made is the C-terminus, or the one that ends with a carboxyl group. Okay? So this is where new amino acids are constantly being added in the C-terminus. Okay? So new amino acids are added there during translation. So here we have a long chain of amino acids forming a polypeptide. So it's also numbered. So the first amino acid is number 1, and you can see that it's found at the amino end. That's the first one that's being made during translation. And the last one, which is amino acid number 127, is also at the bottom, and it's at the carboxyl or the C-terminus end of the polypeptide. Then, because there are certain portions of the amino acid that are actually polar in nature, secondary structure of proteins are also possible to be formed. Okay? And this is due to the formation of hydrogen bonds between the different amino acids along the polypeptide chain. So, one possible conformation of the secondary structure is the formation of a helix, okay, as shown here. You can see in this helix structure that all of the R groups or the side chains of the amino acids are all projected towards the outside of the helix, okay, towards the outside, while the hydrogen bond that keeps the helix as it is in the same plane as the length of the helix, as you can see in these illustrations here. So the length is up and down in this case, and the hydrogen bonds are also in the same plane, up and down. And the R chains or the side chains are all projecting towards the outside of this helix. This helix, because it was first discovered as a secondary structure, is called the alpha helix structure. Then again, it was discovered that some of the polypeptide chains have a different type of secondary conformation. Instead, in a flattened, corrugated manner, as shown here on the right. Okay, and this was discovered later, and this is named the beta-pleated sheet. Okay, and you can see here again the hydrogen bonds are between the two columns or strands of the polypeptides. And the R chains, or the side chains in this case, are projecting either upwards, above the plane, or below the plane. Okay, so you have here your R chains. So again, what's the difference? In an alpha helix, the R chains are to the outside of the helix, while in a beta-pleated sheet, the R chains are either above or below the plane of the sheet. So these are the two secondary structures of proteins brought about by hydrogen bonding between certain amino acid residues. So here's an illustration of an alpha helix. Observe the positions of the side chains projecting outwards of the helix. And here's a representation of a beta-pleated sheet. Observe the folding of one polypeptide so that it appears like it's two strands, but it's actually just one. The R chains or the side chains are not shown in this illustration, and they are supposed to be projecting upwards or downwards.